Hello and welcome to Mishak Milim, which is Hebrew for game of words, play on words. My name is Shaka. This conversation today is with Nicholas Wade, who wrote a fantastic essay about the origins of COVID. We recommend you go and read it. Everything is going to be linked in the description. This was a taboo topic a few months ago. Facebook, if I'm not mistaken, today or yesterday, reversed their policy to remove any content that claims that COVID was originated anywhere else than in the wet markets of Wuhan. And apparently Facebook was wrong, and now they're changing their policy. It just gives you some clue and some insight into the complete, how should I say it, arrogance, hubris that Facebook has been taken here to suppress speech, to suppress this debate on stuff that we didn't have all the data. Not everything was available, and only discussion can bring things to light. And Facebook were not the only ones. YouTube were the same, and Twitter were doing the same. I don't know who exactly reversed what by now, but they were all suppressing this. And Nicholas Wade wrote this essay as a bold step. Unfortunately, it's bold now to go and search for the truth, and he did it in a tremendous fashion. I will let him speak. This episode gets a little technical, but I think it's at a level that everyone can follow, and give us feedback if you think otherwise. A lot of links in the description. I will say just one thing. Towards the end of this conversation, I am referencing something that happened in China, where they stopped domestic flights and kept the international flights going. So there is a small correction there. I'm going to put that in the links. It's not completely clear what exactly happened there. The flight records are contradictory. So the flight plans look as if international flights went out from Wuhan, but not exactly. Some of them were canceled, but they were still registered and all that. I'll let you read through it. So my claim there is not completely accurate, or at least not verified as accurate. So I just wanted to say that as a correction preamble beforehand, and you can read the specifics in the links. Okay, so without further delays, I'll give you the conversation Zohar and I had with Nicholas Wade. All right, it's time to play the game.
Time to play the game. Time to play the game. It's all about the game and how you play it. All about control, and if you can take it, all about your debt. And if you can pay it, it's all about pay. Who's gonna make it? I am the game.
Hello and welcome to Mishak Milim, which is Hebrew for game of words or play on words. My name is Shaka, and I'm here with Zohar, my brother and co-host. How's it going?
Shalom Shachar. What's up?
All good. I want to remind you that this is the podcast where Zohar and I are making an attempt to sounding smarter than we really are, but never succeed. And if you'll think about it, it can be interpreted both ways. So just think about it later. Today we are hosting Nicholas Wade. Hello.
Hello.
How are you? I hope everything's...
I'm fine, thanks. It's good to meet you and see you over the Zoom.
All right. Thank you for joining us. Yes, over Zoom this time, but maybe next time it'll be different. All right, Nicholas, we have a lot to talk about, not a lot of time. You wrote a fantastic piece on Medium regarding the origins of the coronavirus, COVID-19, SARS-CoV-2, however you want to call it. I know different names have different meanings, but we'll go through it. But before we do, because there's a lot to unpack and uncover there, I want you, if you can, just present yourself, give a short, not a bio, but let us know who you are exactly.
Well, I'm a science writer. I've worked on Nature in London, and then on Science, our American competitor, in Washington. And then for most of my career, I've been at the New York Times, where I was an editorial writer, and then I was a science reporter and an editor of the science section. And I retired from the New York Times several years ago and have been writing books.
All right. And you have this Medium blog. Can I call it a blog? Medium page?
No, it's not really a blog. It's just a sort of self-publishing platform.
Yeah.
And that's where I put this piece after I'd tried to interest several people, both on the left and the right of the spectrum. And in any case, by that time, I knew so much about this subject. The article in my head was much longer than any newspaper would really take.
Yeah. So the article.
Obviously, we'll put a link to the article. So you're all invited to go and read it. It's a fascinating read. I want to ask Nicholas, what is your background in science? How did you get to this field? How you become a scientific journalist?
I read science at Cambridge University, but I never wanted to be a scientist. I just wanted to know about it. So I guess I was always headed into journalism, and nature was my first job.
All right.
Got it. All right. So getting closer to the topic at hand, to the article itself, what made you... First of all, I would say it's probably a controversial topic, but a very important one. So I give you praise for even attempting to go there and writing it, and kudos to you. But what made you want to write such an article at all? You know, I assume it was months of work.
Well, it started when I read this piece by Yuri Dagin, which I mentioned in the article. So he's a biotech entrepreneur, and he just put out this beautiful piece. It was also on Medium, that sort of showed how you could take the virus apart and how it might have been assembled. So he didn't say it was concocted in a lab, but he sort of showed how it might be possible. So that appeared in just over a year ago, in April 2020. And having read that article, I then sort of kept reading bits and pieces and trying to sort of follow the track of the evidence wherever it lay, and was increasingly surprised that no one else had picked up on Dagin's piece. Yet there it was a roadmap. It laid out the things you should explore if you wanted to try and find out where the virus came from. But strangely, no one took any interest in it. So it was a welcome distraction from writing a book, which is hard labor. So eventually the germ of the article grew in my mind until I felt I knew so much, I just had to write it all down.
All right, good. So I want to remind our listeners that March 2020, around that time, let's say, even January 2020, we started hearing about that new virus in China, and it came from the market and bats, and suddenly everyone knew what a pangolin was, right? And we took the population at large, right? Took a lot of things for granted. This is what caused it. If they only had sanitation there, that problem in China and all that, and then when it got closer, then it was all that more real and closer. But I think that generally the public conversation was about that wet market scenario.
We all got this meme with the bat in the soup or something like that. We all saw this
memes and all that we took for granted. Someone said a thing, we don't know who that someone is, and we believe it. I'm saying as a general population, that is the narrative, and it's it's an observation showing how narratives really spread, and with no additional evidence, right? And that is now a year later, 15 months later, we're still somewhat stuck with that narrative, even though it has very little ground to stand on. And your article, your piece, article, just puts, takes every argument and every point made on the side of natural occurrence and I think dismantles it pretty thoroughly. So let's go and dive in. I think we should start with some definitions and basics, okay?
Yep.
So because it's going to be maybe slightly technical, as your article was, I think, to a very tolerable degree. But let's talk about it. There's a thing that is important to understand. That's gain of function research, GOF, gain of function. Can you explain it a little bit in your own words?
But it's something that's been going on for about 20 years. And with the new techniques of genetics, virologists can take a natural virus and they can enhance its properties. They can make it a more dangerous pathogen. So you might ask, why on earth would anyone want to do that? Well, the rationale is that virologists can hope to get a step or two ahead of nature, ahead of what nature would do in any case given time, by predicting and hopefully forestalling future epidemics. So we know that we've had 2 bad epidemics already from these bat coronaviruses. And so the idea was, if you could tweak the virus in various ways, you could delineate the most likely route by which it would become a human pathogen. So that is a gain of function experiment. So any experiment that enhances the ability of a pathogen to cause disease.
So there was the SARS and the MERS, the 2 other corona-related pandemics that were, it wasn't pandemics, right? There were pandemics.
Epidemics.
There were just epidemics.
Pandemic,
right?
Pandemic when it's going global. Epidemic
Global,
is
right.
when it's local.
So these are the 2 related epidemics that were in the past, and basically researchers said, let's get ahead of the next virus. Let's make it in the lab, constrained. We can study it. We can understand how it works. And then when it's naturally occurring, we can use our research to deflate it before it's getting big, right?
Right.
That's the idea.
So you're saying, yeah, sorry. So
I'm saying, even let's assume that this is the case. Our success with getting ahead of this coronavirus is not terrific. Let's say we have
so let's ask more generally. So you said it's been going on roughly 20 years, probably around the genome project. What have been the benefits from your background and your knowledge? What have been the benefits of gain of function research thus far, independently of COVID-19?
Right. I mean, in view of the critics, the benefits have been pretty minimal, and the risks have been very large. So I think that probably is overall a fair statement. And there's quite a lot of research being done. One hesitates to say, well, it's all useless, but I think it's fair to say it hasn't produced any sort of big gain. So you can certainly say that the gain so far has not been enormous. And
what's the strongest case for it? What was the biggest gain? Even though you may say it's not big enough, but what was the biggest? If someone would come and say, this is what you don't even realize how terrific this is, we couldn't have done it without gain of function. Is there such a thing that you can think of?
I'm not knowledgeable enough about the field to say. I think virologists would probably come up with lots of little bits and pieces of research saying, it's a reasonable proposition, I guess. If you can put something together, then you really understand it. So we probably do understand quite a lot more about how viruses work, at least from a theoretical point of view, because of gain of function research. But there hasn't been any enormous breakthrough that I'm aware of that has allowed us to predict or prevent an epidemic.
Okay, and are you aware of any gain of function research that spilled over into military industrial complex stuff? Because we've seen movies. If a virus is being manipulated and they're trying to research and know more about viruses, there's the evil general that wants to take it and use it as a weapon. No.
Yeah, there's a constant sort of background chatter about whether there was Chinese military involvement So I've tried to avoid all that because I don't know anything about it. It seems to me you have a much clearer discussion if you stick to the science. And the first question is, did it come from a lab? Did it escape from a lab? Now, it could well be that there was military involvement. And if, for example, the Chinese military had shown a great interest in these viruses, which are, after all, kind of lethal, and if it had tried to develop a vaccine to protect its troops, I think this would, most people would regard this as a routine military activity. This is what militaries do. Now, if beyond that, they'd actually tried to make a bioweapon of it, I just have no information. I just have speculation. Militaries have crazy people just as the rest other institutions do. So there could have been some branch of the Chinese military that wanted to explore making a weapon of these coronaviruses. I mean, most military people, as I understand it, really do not like chemical or biological warfare. These are ugly, messy weapons. They're hard to control. Most officers would much prefer to have, another 100 tanks rather than a lot of people spreading bioweapons. So I just don't think it's sort of useful at present to explore that when we have no data on which to do so.
All right, I want to reinforce what you just said, and I wanted to bring that topic up, but I think it's the easy escape. Because obviously, first of all, it makes things more complicated and you need more evidence and it's confidential and you're throwing a lot more politics in there and you should avoid it in your essay. But regardless of that, that makes it, evil. And then, we don't want evil things, so we're going to be against such a thing. So, okay, but I think that the arrogance of doing it even without the evil general that comes in and wants the weapon, the gain of function research itself, without that military usage per se, is on its own something that just invites the evil general or the evil terrorist or a negligent doctor, right? So I want to talk about the harder thing and the harder reality of doing those things and playing with those things without the evil general. So if we go back to movies, right? If you remember the movie Alien, right? It's a classic. If you haven't seen it, then spoiler alert. But they take the alien, they take that larvae and everything, that parasite, and there's someone, the science officer is actually an android, is trying to harvest that alien, trying to get that alien to research it and maybe use it eventually, right? To gain of function on the xenomorph. And I think that the human race, regardless of military usage, the human race would have done the same outside of the movie realm. We would have taken that xenomorph and we would have tried to research it and tried to control it and see how we can defend ourselves from the other xenomorph species and all that. We would have done it. How do I know? We are doing it now. We're doing it in gain of function. We're doing it with CRISPR. We're doing it with other stuff. I'm not arguing that I know what to do. I'm just saying this is arrogance. And we should just be aware that we're arrogant as a species. and maybe put some more guardrails if we can. Maybe it's a too strong statement for some, but that's how I see it. All right, so gain of function, its objective is to learn more, be prepared for future viral infections. We'll be more ready with vaccines and all that. That's the holy grail, right? We'll know how to make vaccines for viruses, right? But in order to do that research, viruses are taken into a lab and enhanced, correct?
Yes, I don't know that all vaccine technology depends on gain of function, but certainly some of it might do.
So a part of the research, again, some of those are the places that do gain of function, they enhance and make the viruses more inclined to infect human tissue, make them more adaptable to humans. That's part of the thing. In order to test it, you need to make it infectious first, no?
Yes, that's right. I mean, you tested it on humanized mice, not on human subjects, but yeah, that is the goal.
All right. So that's
so in nature, there are a lot of coronaviruses. It's a whole family, and there are a lot of them in bats, and they can't infect human cells most of the times, right? And what they do in the lab is they try to see what kinds of manipulations the viruses can go through that will cause them to infect human cells, and this is what they're researching. Am I correct?
Yes, that's right. You start off with a particular coronavirus, and then there are various things you can do to it to make it more infectious. You can drop in the gene for a spike protein from a different virus that may have a better affinity for human cells. You can insert this thing called the Furin Cleavage site that makes human cells much better target. There are all kinds of things you can do to soup up the virus, to enhance the virus's ability to infect humans.
So can you describe just in a couple of sentences, what do we know about how SARS-1, the 2002 epidemic, and MERS, how did they came about? Just again, I know you're not the one that made the entire all the links in the chain, how it came about, but there's general knowledge on how this worked back in 2002. Can you describe it?
Well, in the case of SARS-1, which was the 2002 epidemic, we know that it jumped from bats to civets, which are kind of animals sold in Chinese wet markets. And from civets, it jumped to people. And you can follow the mutational changes as it made these jumps. It would acquire one mutation after another, and each mutation made it sort of better able to affect civets, and then better and better able to affect humans. And when it had accumulated, I think it's something like 32 of these mutations from its sort of bat form, it then became a very fierce human pathogen and set off this epidemic. So the case of MERS is not quite so clear cut, and I think I might say we're not really sure of the bat population for the virus found its way to camels or dromedaries. That was the intermediary species. But once it was in camels, it then again made sort of several jumps to humans until it sort of perfected its ability to infect human cells and set off that epidemic. So
there is a genetic trail of crumbs, if you will. I don't know if I'm translating it correctly, but a track record genetically that we see the different mutations and how it's slightly changed from step to step, how it came from bats, infected maybe some species in the interim, and then made the adjustment to humans. And even with humans, there are more adjustments and more changes that make it potent and dangerous. That's what we've seen with both SARS-1 and MERS. And I think, again, I haven't seen I don't know how to read the stuff, the genetic track record, but it's pretty obvious that that's the case, right? As you said, 30-something mutations. We see the graduality of it. This is evolution at work. You can you can argue this is not natural evolution, but there is a sequential process, step by step, that has no argument at this point in the scientific community.
There are traces that we can that we could identify almost 20 years ago, back in 2002, and since then. That right? Is that correct?
Yeah, that's right. And there are lots of trails of crumbs all over the place. This is natural evolution, but it leaves copious fingerprints in the environment. So if you suspect you have a case of natural emergence, you can go back and look for them in the serology of the human population, because hospitals keep all these surveillance records that record what pathogens our population has been exposed to. You can look for your trail of crumbs there. You can look for your host, intermediate host animal, and you can try and find the source bat population from which it all came. So there's lots of evidence waiting there to be found in the case of natural emergence.
Okay. And another general statement that I will say is that evolution is very effective, but not necessarily efficient. So, for example, the human body, the main arteries to the lungs and to, that they're intertwined, they're not direct routes. There's so much real estate that is wasted because it wasn't planned efficiently. It just, the mutations, again, hypothesizing, the mutations just happened gradually, and what worked, and so it was effective, not necessarily efficient. So in a situation of natural occurrence, you see effective things, not necessarily the most efficient way to infect a human, but it does work, but it may not be, the easiest way. I'm saying this general statement because we'll revisit it, I guess, in 10 minutes or so. Okay, so by the way, the SARS virus, the SARS-1 virus, is still very close to the origin that came from bats, right? And bats are somewhat susceptible for it, or at least we know what makes it susceptible for bats, right? The backtracking is pretty straightforward. We know the human version of it, we know the bat version of it, and we know how it came from one to the other and what relation it has to bats and infecting bat tissue.
Yeah, there's a pretty complete story of it.
All right. Background finished. Let's talk about COVID-19. So, as we all remember, it started in Wuhan, in China, and the wet market story came out first minute, or even before we really noticed what's going on. It came to our attention even 15 months ago that there is an institute in Wuhan as well, but it's a conspiracy theory to think that they did anything like that because they wouldn't do it on their own people, or something like that. And also, blaming China was considered siding with a certain president, I guess. But we can leave the politics aside. So right next to Wuhan, there's this Virology Institute. Can you speak about that institute just a little bit? And we'll see what they did there and what information is known to us at this time.
Well, the Wuhan Institute of Virology is the leading lab in China for the study of coronaviruses. So it's headed by a well-known bat virus scientist called Dr. Zhengli Shi, who has the moniker of Bat Lady in China because of her long-standing interest in these viruses. And it produces scientific papers just like any other lab does in the world. And that's how it came to be on the map.
Is it on its face, sorry, Zohar, on its face, above ground, declaring that they're doing gain-of-function research as well?
I don't know if they've ever put out any public statement to that effect. Dr. Shi gave a long interview with Science, but I'm afraid I've forgotten the details of whether she addressed that point.
Okay, I think her partner did. I'll search for it in your article.
I want to ask regarding geographically. So obviously the Wuhan Institute is close to the Wuhan city and to the wet market. That is to blame for this epidemic. But the bats themselves, where are they located? Are they close to the lab? Are they in a different place? What's the geographical proximity there?
So these bats hang out in caves in Yunnan, which is in southern China, and that's about 1,500 kilometers away from Wuhan. So,
which is a lot in Israeli terms, that's for sure.
Yeah, that's Italy, I guess. That's Italy or something like that.
Yeah. It's three times Israel.
Yeah.
It's a big distance. The bats are quite widespread over China, but individual bats don't fly very far. And it seems very unlikely that a bat itself would have directly taken the virus to Wuhan, because at the time the outbreak occurred, which probably was in September, not the official version of December 2019, prevailing temperatures are quite cold and bats go into hibernation and such. temperatures. So the fact is that if the epidemic had sort of broken out in the caves around Wuhan, it would look exactly like a natural epidemic. It had jumped naturally from bats to humans. So it's very hard to explain how you get the pandemic to break out somewhere in China, not in Wuhan, and sort of get it to Wuhan without leaving some trail. So do you have one guy infected? He somehow doesn't infect any member of his family. He jumps on a train to Wuhan, he gets off, and there the virus starts to spread. I mean, I guess it's possible, but it's just a very sort of strained explanation. Whereas if you think it was lab escape, you know, you've got the lab right there, right where the epidemic occurs. So sort of common sense tells you there could be a connection.
So I think it's an important note to say that whatever we say here, whatever you say here, you don't claim to know the answers. You're just saying a lot of the things are much harder to explain with a certain hypothesis. It's not that it's clear that it's not a certain theory, but it's much harder to explain it with the certain theory. So
yeah, I think that's exactly right. That you've got these two plausible scenarios. There's no direct evidence for either one. But if you then take the available evidence and say, well, which scenario explains the evidence better, I think on a whole range of points, you find that lab escape gives you the much easier explanation. So that's why my conclusion so far is that on present evidence, lab escape is the better explanation.
Okay, right. So let's go through some of these points then.
Wait, I want to read something we didn't remember before, so I'll read something from your article. On December 9th, 2019, before the outbreak of the pandemic became generally known, Dr. Daszak, I don't know how to pronounce his name.
Peter Daszak. Yeah.
Peter Daszak gave an interview in which he talked about, in glowing terms, of how researchers at the Wuhan Institute of Virology had been reprogramming the spike protein and generating chimeric coronavirus capable of infecting humanized mice. Quote, and we have now found, after 6 or 7 years of doing this, over 100 new SARS-related coronaviruses, very close to SARS. Some of them get into human cells in a lab. Some of them can cause SARS disease and humanized mice models, etc., etc. So this is a partner to Dr. Shi, correct? To the bat lady. He's a close partner of hers. They, if I remember correctly, they submitted grant requests together. So he's declaring in an interview that this is what we're doing, reprogramming the spike protein of coronavirus. It's December 9th, 2019. This is just days before the pandemic started. Okay? So that's revisiting the question of whether they ever declare that that's what they're doing. I think this is as close to a declaration of about gain of function research as you can have. All right. So the distance, okay, 1,500 miles, but there is this plausible scenario where maybe, maybe it's a hybrid model. Maybe a scientist from the Wuhan Institute of Virology traveled, took bat samples, guano and everything, came back to the lab because they're doing the research there, and that scientist or scientists were infected, so they are patient zero. We don't know who they are, but it's natural occurrence, but they just happen to be from the lab because they're traveling to that Yunnan province all the time. That is a plausible scenario, plausible solution. Maybe not plausible, but a possible scenario. So we need to dissect other characteristics of what's going on in order to see if that is plausible. So one area that we need to dive into is a little bit of the science and the genetics of the virus. So it appears like, as we described before with SARS, there were 32 mutative steps that had to be undertaken. Here, it's not visible that we have a backtrack at all. Can you expand on that a little bit?
Well, we have a kind of a backtrack in that very soon after the full genomic sequence of the SARS-CoV-2 was published, the Chinese came out with Dr. Shi came out with the sequence of another very interesting virus called RATG13. So this virus performed an important function in that it gave you a sort of plausible bat ancestry for SARS-CoV-2 because it was sort of quite close to SARS-CoV-2, but not close enough for anyone to say, well, this is the origin of SARS-CoV-2.
Where did we see that virus? In an article she published?
Yes.
So that is explaining her research. This is what we found. This is a virus that we found, and probably explaining a lot about that virus and ways of treatment or whatever she was doing, right? That's the purpose of the article. That's what she wrote.
Yes, it sort of fits in with the preconception that this was a natural virus that came from bats, because here is something halfway between some putative bat population of viruses and SARS-CoV-2. But there are lots of very curious things about this virus and how she published it and why she originally published it under a different name and why it's almost impossible to reconstitute that viral genome from the data she's put in the public record. So there are a number of clues that suggest that this is a concocted virus and it's not a real sequence. It's something she has placed there in the public record to make it seem as if SARS-CoV-2 came from a natural population of bats.
Can you just specify a few of those suspicious elements? I never knew she published it under a different name. It's pretty wild. But what other what other clues? If you can expand in a minute or two. Okay,
well, she first published it under the name of BAT-CoV-4991. And this, it was a fragment of the virus, and it came from a cave in which a number of miners had been working, digging out bat guano, and 3 or 6 of them all died from exposure to a virus in that cave, which seems to have been very SARS-2-like, according to the pathology. Now, because this was a lethal virus, it would seem Dr. Shi did not wish to establish a connection between 4991 and the virus she was publishing, RATG13. So she renamed it. We know she renamed it because the fragment of 4991, it's just one gene. It's almost identical to that of RATG13. Mm-hmm. So firstly, she renamed the virus without saying she'd previously published a part of it. Now, you're not meant to do that in virology. It's very bad form. It's deceptive. So that is the first hint that something is wrong with or something is odd about this viral sequence. And the second is that when you publish the sequence of anything, you not only do you publish the sequence in GenBank, in these various genetic databases, you must also publish all your raw data. Every bit and piece that comes out of the DNA sequencing machines, you must also publish. So if you take Dr. Shi's raw data and you try to reassemble RATG13, it's very difficult to do it. In other words, you cannot see how she got from A to B. So that too is not good science.
Okay, so suspicion arises, and you're saying that virus is close. It looks like middle ground between what we know in bats and what we kind of middle ground between the bat viruses and SARS-CoV-2.
Exactly. Yeah.
Okay. So, all right, that's one thing. What about in your article you specify a little bit about spike proteins? Can we discuss that? Spike proteins, and I think, Zohar, maybe you would have more to say there, but those are the agents or the parts of the virus that are responsible or make it possible for a virus to penetrate and infect a cell in the host, generally
and I think
speaking.
that we can get technical here, Nicholas. I mean, I hope that the listeners can follow, but let's get technical because it's interesting and it's important.
Okay, sure. So the spike protein has two functions, and it also has two parts that are called S1 and S2. So S1 is the part of the virus that recognizes the target cell in the human airway, which is a protein called ACE2. Now, other animals also have the same protein, but in different forms. So the spike protein is very important because it determines what host the virus can attack.
Let's dive in for a second. This is all chemistry, chemical bonds, right? So yes. The spike protein is just based on its structure, on its components. It creates chemical bonds with certain types of cells, certain types of other proteins, I assume, and it's going to be specific. It can't go on my skin and infect my skin, probably. I don't know specifically, but for the sake of the argument, but it does have it's an expert in recognizing, creating chemical bonds with the airway tissue of a human host. That's what you just said on S1, right?
Yes, that's correct.
Okay.
So this is the first part of the spike protein. The S1, it has what they call the receptor binding motif that recognizes very precisely a bit of a protein molecule on human or other cells. The protein molecule is ACE2, and depending on the exact sequence of amino acids in the receptor binding motif, the spike protein will attack bats or cats or mice or humans. It's all very specific. And then the other part of the spike protein called S2, that is the one that helps it merge with the membrane of the cell and drags the virus into the cell so it can inject its nuclear material and hijack the cell's protein, DNA making machinery, and order it to make more viruses.
So S1 creates the bond and maybe even creates a tunnel. I'm just making it visual, okay? I don't know if it's exact, but it creates the bond, connects to it, and now there's a tunnel where S2 goes in and does its stuff, does its thing.
That's
something
right.
like that.
That was exactly a tunnel. I don't know if anyone has visualized it, but S2 certainly does merging with the membrane.
Mm-hmm. That is the thing that penetrates the membrane. S2.
I think what happens is that the membrane of the virus sort of merges into the membrane of the cell.
Diffuses, yeah. Mm-hmm.
That they fuse, and then the virus's genetic material, which happens to be RNA, not DNA, is then injected into the cell.
Okay, so that is pretty common, right? S1, S2, this is not uncommon spike protein description, correct?
Yeah, all these coronaviruses have these spike proteins.
Okay, so what in this case stands out?
Well, what stands out is that for S2 to do its thing, it needs to be cut away from S1. So the virus does not carry its own cleavage thing. It's a very sort of economically designed piece of RNA. It relies on the victim cell to make this cut. So all human cells have on them, not for this purpose, obviously, but for another purpose, an enzyme called furin, which will cut proteins whenever it sees a particular sequence of amino acids. So if you want a protein to be cut by furin, you need to have in it this particular site recognized by furin. It's called the Furin Cleavage site. So if you look at the structure of SARS-CoV-2, there, the S1, S2 junction, just where you need it, is a furin cleavage site.
Okay, so cleavage means a split, right? Something that splits, and furin is the material. So you're saying furin cleavage is a chopping knife. That's what it does. Furin cleavage from now on. So So you're saying that the virus itself has a furin cleavage, or
Yes, in its genome, it has a sequence of RNA nucleotides that specify the amino acids in the
yes.
spike protein chain that are the furin cleavage site.
Okay.
So in order for the S2 segment to work more efficiently, it has to be cleaved. It has to be cut from the S1. And what's special about this furin cleavage site? Why is this something that caused suspicion? I mean, I assume that in natural viruses there is also some kind of cleavage site, and the viruses are quite efficient. So what raises the question about this particular cleavage site?
Okay, so there are furin cleavage sites possessed by other viruses, but in the family of viruses to which SARS-CoV-2 belongs, they're called sarbecoviruses. No other known member of that family has a furin cleavage site. So that raises the question of how did SARS-CoV-2 acquire its furin cleavage site? And this is something of a problem. So viruses sort of gain new powers in two ways, either by mutation, like SARS-1 did as it sort of developed itself into a serious pathogen, or by recombination. So that is a swapping of genetic material, and it occurs when two viruses happen to infect the same cell, and they get their bits and pieces mixed up as they are assembled in the cell. So by recombination, you can acquire a lot of new properties. The one thing you cannot do is acquire a property that is not present in the gene pool of your viral family. So that is why it is a real problem on the natural emergence theory to explain how SARS-CoV-2 came by its furin cleavage site. On the lab escape theory, it's easy. It's well known by virologists. If you really want to enhance a virus's ability to attack human cells, you insert a furin cleavage site. So you can see in the literature at least 12 papers reporting doing just that to a virus.
Can I try to clarify that point for maybe someone like me that is struggling to understand this? S1 and S2, the spike protein, they are one chain of protein, right? They are one genome,
sequence.
Okay, yeah. Sequence. And they need to do the following. They need to connect to human tissue, disjoint themselves from each other, and then S2 goes in, does its thing, right? So three-step process. What happens here is that once S2 is connected to the human tissue, S1 and S2 are still connected, but with the connection process, there's this diffusion that you said, and that diffusion actually brings some of the, the chemical bonds actually go and cut the furin cleavage site precisely at the end of the S1 protein, because this is the furin cleavage site is very adapted to the human tissue to do what it normally does, right? This is something that is known in human tissue, furin cleavage. This is precisely, again, I don't know, maybe I'm saying words that are not accurate, but humor me for a second. The human tissue knows this sequence of the furin cleavage, that's a few amino acids one after the other. It knows it, but the function that it does, it cuts it, and that basically releases S2 away from S1 to do what it wants to do. Is that right? What you're saying? So,
yeah,
so the furin cleavage is...
But it's not specific to human tissue. It also occurs in other viruses. So
no,
this is not...
But it's different cleavage sites, right? This is what I'm trying to understand. The furin cleavage, it's a specific sequence that may be slightly different with different animals. And maybe I'm wrong here, so please explain what I'm not getting precisely.
No, the furin cleavage site is always the same, because furins, I think I'm right in saying all furins will cut at this particular site. What is different is viruses use different means of entry into their target cells. So bat viruses don't attack furin to get into bat cells. They have another target sitting on bat cells that they use to affect their entry.
So what's interesting here is, if I understand correctly, is that all the coronaviruses, the entire family, doesn't use the furin cleavage, doesn't use furin to cut it. It uses a different mechanism. It does happen. It does happen with other viruses that they use the enzyme called furin to cut their proteins. But so this is not something out of the ordinary.
It is out of the ordinary in
what's out of the ordinary here? It's that it's in coronaviruses, which and SARS-CoV-2 is the only virus that has this this sequence. The other interesting thing is that this sequence is used by humans to design cleavage sites.
Is that accurate, Nicholas? I think
yes, that's right. I should explain that in this sort of big family of viruses, it's called beta coronaviruses. There are several subfamilies. Now, one of these subfamilies, I didn't mention its name in my article because it sort of sounds so technical. One of these subfamilies is called sarbecovirus. So, and this is the family to which SARS-CoV-2 belongs. So no other sarbecovirus has this furin cleavage site. There are other beta coronaviruses in different families that do have furin cleavage sites. But viruses, by and large, don't combine outside of their own family. Recombination is a rather sort of intimate thing, and you have to have a very sort of similar genomic structure in there to recombine with it. So by and large, viruses only combine inside their own family.
So there are three main hypotheses, three options that a virus could acquire this sequence. The one is mutation, and it's not very plausible because this is a relatively large sequence, and through mutation, it's hard to get the specific sequence. It's like, I don't know, winning the lottery, or I don't know, some other analogy to give there, but it's not
impossible. It's just improbable.
It's not impossible. It's just not very plausible. The other way is recombination, but it's also not likely since it doesn't happen without. It only happens within the family, and there are no other viruses within the family that has this cleavage site. The third option is genetically engineered sequence inserted in a lab, right?
Right.
Okay. So again, everything that we talked about, nothing here is impossible. Makes the natural occurrence impossible. Just it's a compound of improbabilities. The distance and the genetic structure and the lack of backtracking and all that. And I just want to get back to the point of efficiency versus effectiveness. So coronaviruses are effective in penetrating human agents, and I don't know exactly how they do it, but the cleavage site is a very economically effective way, as you nicely put it in your article. It just makes it, it's not effective, efficient. It's very efficient. It's the easiest way to do it, and it's the exact mutation that needed to happen exactly there, and it's unseen in other coronaviruses the way it is like this. So not impossibility, but just improbable is a mild statement. Okay, so now we talked about geography. We talked about the technicality of the biology of the virus. I want to talk about the institute itself. I think it'll be the last point. So we imagine when we're talking about a institute of virology, I know how it looks, right? People in spacesuits, masks all around, gloves. Rooms that are completely sealed and airtight, purification procedures as people go in and out, etc., etc. Because, you know, again, I've seen movies. What can I do? This is how we get a lot of our impressions and how things work. So I've seen it. And to think that a virus would come out of that secure location seems improbable to me. And maybe it's an uphill battle for you, Nicholas, no?
No, it should be improbable if the experiments had taken place in such a lab.
Okay.
But they didn't. They all took place in labs of much lower biosafety levels.
Okay, how do you know that?
We know partly because Dr. Shi herself said in an interview with Science that all her work was done in BSL-2 or BSL-3. There are 4 levels of biosafety, BSL-1 to 4. So the 4 is the highest, and that's the spacesuit air purification that you mentioned.
Yes.
So all her work was done in BSL-2 or 3, according to her own statement. Her own papers describe mostly work done in BSL-2. And you should understand BSL-2 is not very high level. You put up a biohazard notice on your door. You wear a sort of coat and gloves. You do some of your work under a hood, but it's not very high tech stuff.
So can I say a sausage factory has BSL-2?
Well, the analogy
roughly
given is a dentist's office.
Dentist, yeah. Yeah, dentist office has BSL-2. So if you went to your dentist or maybe to your doctor, it's probably BSL-2 or very close to it, right?
You wouldn't work with any
no spacesuits,
lethal viruses there, I guess.
Okay, and BSL-3 is a big leap.
No, the big leap is between BSL-3 and BSL-4. So BSL-4 is used for seriously dangerous things like Ebola.
Yep. Okay, so that Institute of Virology has worked on BSL-2 and BSL-3 safety levels. Again, bio security levels, I think, BSL, right? So doing such a thing with coronaviruses that you're doing gain of function research, making the viruses more adapted to humanized mice, which I don't know if we can get around to explain what that is exactly, but mice that are made more closer to the human biological characteristics, I will say. So all that's playing with fire. But I think there was also a report from probably not the WHO. I think it was the U.S. Embassy that sent inspectors to that institute in 2018. Can you remind me who that was exactly?
Yes. So this BSL-4 lab, it was built under somewhat uncertain circumstances by the French government for the Chinese. So it's a very high-tech lab. I think the French expected to be asked to run it, but I understand the day they finished it, they were all asked to leave. For some reason, the officers at the U.S. Embassy in Beijing decided to inspect the lab, and they reported back to the State Department that it was seriously deficient in its safety procedures. It was not being run correctly. Many of the things they did were inadequate.
Mostly shortage of trained technicians, if I remember correctly.
I will quote from your article, quote from the U.S. Embassy inspectors. The new lab has a serious shortage of appropriately trained technicians and investigators needed to safely operate this high contaminated laboratory. That's one. And besides that, they said that, or we saw in other places that what should be BSL-4 or 3 is operated in BSL-2, etc. Okay, so maybe not best practices. Okay, I get it, but can we talk about what was maybe the involvement, or what was the involvement of the U.S. in that institute, and where did the money come to that research and all that? Because I think it does add color to the situation. Granted, it should add a lot of noise. I don't want to add political noise here. I just want to give the full context and background on what has been going on. But what's the involvement of the U.S. in this?
Well, strangely enough, Dr. Xi's research was funded, at least in part, probably in small part, by the National Institutes of Health in Bethesda, Maryland. So the NIH gave a grant not directly to Dr. Xi, but to Dr. Peter Daszak, who we've mentioned, who is president of an organization in New York called the EcoHealth Alliance. So Dr. Daszak is what they call the principal investigator, and he then subcontracted the funds to Dr. Xi for her to explore the range of coronavirus infectivity.
Yeah, okay. So NIH, National Institute of Health in the U.S., and there's another institute called NIAID, Natural Institute of
Allergy and Infectious Disease.
Yeah, thank
That's
you.
one of the sub-branch, one of the component institutes of the National Institutes of Health.
Okay, and if Israelis here think about the coronavirus project manager for the government, the NIAID is the one responsible for that. That is the project manager for the U.S. federal government. The name that is on everyone's lips is Dr. Anthony Fauci. He's the head of that institute, I think, since the 80s, which is crazy. But he's there, and he's been the face on the screen for 15 months now.
And let me just say that we know what the NIH funded, right? What is the grant? What is the research proposal? It's I'm not sure about the specific, but they claim to research preventions of pandemics. That there is no question that they are researching viruses, SARS virus, COVID virus, not COVID, coronaviruses in this lab in Wuhan. That's not a question, right?
Not a question. No.
Okay, so just 11 days ago, Dr. Fauci was in a Senate hearing, and he said that the U.S. government was not funding gain-of-function research in that facility in the Wuhan Institute of Virology. So, what's the disconnect there? Can you explain that?
Okay, this is a very complicated issue, and we don't yet understand the bottom of it. But from what I've got so far, I think the situation is this, that the US government certainly did not intend to fund gain-of-function research at the Wuhan Institute. Even though the grant abstract strongly suggests that gain-of-function research would have had to take place, nonetheless, that there are some experiments you put under that description that would not result in a more dangerous virus. So let me explain that a little, since it's so kind of counterintuitive. Supposing you took the backbone of a coronavirus, which had somehow been weakened or attenuated, as viruses say. So you've got this very weak backbone, not at all good at infecting human cells. So then you take the spike protein from another coronavirus, and you want to see how good this spike protein is at infecting human cells, or probably your humanized mice. And you drop this spike protein, or the gene for this spike protein, into your attenuated background. So now the question is, would this new chimeric virus you've called, a chimera is when you meld the genomes of two different viruses, would this new chimeric virus in fact have a gain of function with respect to the two component viruses that made it? So if you've got the picture, you've got your attenuated background coronavirus, and you've got the spike protein from another coronavirus. Now, you could argue that because of the attenuated background, this new chimeric virus you've made would not have a gain of function. It would still be unable to infect human cells in itself.
Okay.
So you see this rather narrow line that is being drawn here. So someone else could say, well, how can you be sure about that? You're creating a novel virus. How can you'd be so confident you can sort of predict all its functions. And moreover, the overall purpose of this program was to assess the infectivity of coronavirus spike proteins for human cells. So how can you be sure that in the course of this program you wouldn't produce a virus that did have a gain of function?
Okay,
so the answer to this debate lies somewhere in the precise grant proposal. Remember, at present all we have is the abstracts. If we could see the whole grant proposal, maybe this would convince us one way or the other. But the bottom line, I think, is that you can say that the NIH did not intend, it took precautions to make sure it would not be supporting gain of function. And yet, it seems certainly from Dr. Dasak's description at his interview, which you referred to, and all the history of what Dr. Shi had been doing in making chimeric viruses, that this was the direction the research was definitely taking.
Okay, so to be to have good faith towards Dr. Fauci, either it was not intentional, right? So basically, he says we didn't fund this. Maybe they did something we didn't fund, right?
Yes, yeah, that could be a possibility.
Yeah, that is a plausible interpretation. I think another plausible interpretation is that the money didn't go to the Wuhan Institute of Virology. It went to Dr. Daschak, or however again you say his name, Peter Daschak. And we just funded his research, and he just happened to do it in a certain virology institute with some partner who's a bat lady. So if you look at it like that, we never funded the Wuhan Institute of Virology gain of function. We just funded this guy. I think it's another plausible interpretation to say this is not a lie. Yeah.
That's certainly a question, but the NIH is not taking that route. It's not saying we funded Dashak and he did his thing. It's saying specifically, and in fact, people who receive grants from the NIH don't have that much freedom. They must do exactly what they said in their grant proposal. So the NIH is saying this grant proposal was written in such a way you could not do gain of function research.
Yeah, I get it. That's fine. And I did not read the NIH's response. I just listened to the under oath statement of Fauci, and he said specifically, he repeated the following words, and it's not a direct quote, but he kept saying no gain of function research in the Wuhan Institute of Virology. So he kept saying this entire statement under oath together, right? So I'm saying, okay, he's taken either route. Either it's not gain of function, or for me, right, this is my interpretation, or it's not in the Institute of Virology, because he made sure to say the Wuhan Institute of Virology every time he addressed that issue, and that's under oath. So again, I don't know what the NIH said. I don't know what's true. I'm just giving another possible interpretation. But okay, so just to clarify, we are seeing, and we'll put a link here to the grant abstract. You're saying the abstract is all that's exposed to us, to the public. There's no full dive. We can't dive into the grant itself, just the abstract.
Yeah, these grant proposals are always kept public, I guess, to preserve the researcher's sort of right in his own intellectual property. Got it.
Okay, that's interesting. That public funds. It shouldn't be. I don't think it's security issues, security secrets, confidentiality. So it's weird that public funds are going to something that's not exposed. All right. I think this covers. So we've seen improbabilities in geography and genetics. Any other things that that you want to cover in this thing, just to explain what's the probable source of the SARS-CoV-2?
Well, I think one of the oddest features of the virus. Do you want to have a break? Okay.
No, it's fine.
Okay.
You can continue.
All right. So one of the most remarkable features of the virus is that right from the start, it was very well adapted to human cells. So in this way, it differed from SARS-1, which, as we've discussed, took a long time and the acquisition of several mutations to sort of perfect itself as a human pathogen, this thing right off the bat was very good at attacking human cells. And moreover, it hasn't changed very much since, apart from these very recent variants that don't seem terribly significant. The virus has stayed the same. So this is very, very peculiar and very hard to explain on the natural emergence theory. Moreover, if you look at the virus's proclivity for various different types of mammalian hosts, it binds far better to human cells than it does to bat cells. So this is very odd for a bat virus that hasn't changed to prefer humans to bats. You have to scratch your head and say, what on earth is going on? Hard to explain on natural emergence, but very easy to explain on lab escape. These viruses were being trained up on humanized mice, so of course they attack human cells.
Yeah,
and I urge all the listeners to go and read the article. It's really well written, and there are other points there that are that you talk about. Let's say like the arginine codons that are not prevalent in these kinds of viruses, but I won't go into the details of that. Just go and read the article. I just wanted to say that even if you are going for the not the natural occurrence, but going for a lab leak theory, it doesn't mean that it happened in a malicious way. It doesn't necessarily mean that someone planned on it to happen. Most likely, it happened by mistake. Just they did research these viruses, and with some human error, it just leaked out of the lab and started to spread. Now people are trying to persuade us that it didn't happen because of them or because of their research, or it's not related to them. So you can see how the human mind wants to defend themselves and their reputation. But it doesn't mean maliciousness.
As in with many things, sometimes the cover-up is worse than the initial mistake. And to me, it seems like I have no reason to believe there was maliciousness in a leak, which is to me the most probable explanation. But there was maliciousness in the cover-up. And I'll just there are a few examples, and you talk about a few in your essay, Nicholas. But one that I think really stands out is that the Chinese government understood that it has a crisis on its hands, and basically put Wuhan province in aerial blockade, meaning there were no outgoing flights. But that's not completely accurate, because there were no outgoing flights domestically into other parts of China. They completely isolated Wuhan from the rest of China and no outgoing flights. But they left outgoing flights to global destinations. So Wuhan airport, I assume there's more than one, couldn't fly to Beijing, but it could fly to London and to Milan, where the first Western world serious outbreak happened, and to New York and everywhere else. So the Chinese, in order to cover up and to save face, didn't want to stop aerial traffic outbound because it would just be clear that something's up. But they did try to make their population safe with stopping outgoing flights into other Chinese destinations. So this is maliciousness. And the cover-up here is worse than any innocent mistake that may have happened in that virology institute.
I think the Chinese have a lot of accounting to do.
Yes, maybe finally. Nicholas, maybe one more small question, and I think we can wrap up there. But I heard, I don't know, I haven't seen any reference in your article to this, but I heard that the spike protein, or maybe it's a different protein in coronavirus, it has the proclivity to create bonds with more than one tissue type in the human body. So not just airway cells, but also maybe heart muscle cells, maybe other places. Is that something that you've seen in your work, or not really? So it's suspicious that it can connect with more than one type of tissue. Have you seen something like that?
Yes, I've heard that, but I don't know enough about it to say anything.
Okay, so we can leave it at that as something we both heard of, but we can't say that it's true. Okay, I think that's a good place to stop, unless you have any other points you want to make. And assuming you don't, we always finish, as we told you, with some cultural recommendations, looking outside of this topic at hand, and maybe giving people some things to see, read, explore, expand their horizons, and see other stuff. I want to recommend something first to give you another minute to think about a recommendation, and I will recommend a podcast series called The Dropout. It's a series. I think it's 7, 8 episodes, something like that, each around 30 minutes, give or take, and that is about Theranos. Theranos was a company that I think demonstrated herd mentality, demonstrated how we can be convinced by narratives, as we talked in the beginning about the wet market hypothesis, that was just the narrative, the ruling narrative, and that's it. So Theranos built a narrative around itself as a medical biotech company that developed a novel technology, take a drop of blood and we can inspect any disease that you may have, and it's the future is here, this will change the world of medicine and all that, but it was all a charade, all a sham. There was no technology behind it. It was just fluff, aggressive marketing, and a good CEO or a good marketer CEO that just played everybody, and people wanted to believe. And this was a company that was worth billions of dollars overnight and then crashed to nothing because there was nothing there. So Theranos is a story about the human psyche more than anything, but I think it's a fascinating story. So The Dropout, that's my recommendation.
Right. I'll do also a quick recommendation. Now we're watching on Netflix. We're watching Shtisel. So we got a recommendation from our cousin and from our aunt, and it's on Netflix also in the US, and it's in Hebrew and in Yiddish. So it's funny to hear all the Yiddish speak. That I'm able to catch a few words here and there, so it's nice. But there are also, of course, English subtitles, and I know a few Americans that watch it that are not Jewish at all. So I think it's an interesting story. It's well done, and I recommend.
Nicholas, anything from you?
I don't usually read novels. I think I've read so much non-fiction, my mind has been degraded for reading novels. But one I read recently, I greatly enjoyed. It's called Circe by Madeline Miller, and it's the story of the enchantress Circe, whose island Odysseus landed in his travels home from Troy to Ithaca. And it's beautifully imagined. It weaves in lots of Greek mythology. You see all these characters, from myths, are brought to life and integrated in one beautifully imagined story. I didn't really enjoy the passage where Odysseus's sailors are turned into pigs, but they get turned back into men again, so that's not so bad. And it's beautifully written, so I definitely recommend that to anyone who likes that early Mycenaean world.
So it's that part of the Odyssey told from her perspective?
Yes, it's all centered on her and her island, and she visits other places in the world. She goes to Crete and visits King Minos, and she gets around the ancient world and brings all these wonderful ancient Greek stories together in a sort of united theme.
Sounds good.
All right, sounds really good. All right, we'll put a link to that. Okay, as we said before, everything we discussed will be linked. Your essay obviously will be linked first, and then from it you can dive in deep into the rabbit hole, but we'll try to put some things obvious to you here in the description. So you can go and click and see for yourself. Nicholas Wade, I think your essay was of tremendous importance, because if we don't know what happened, how can we improve for the future? So that's a very important first step. And unfortunately, probably a pretty brave step. So we congratulate you on both the achievement and the ferociousness in which you've done it. So thank you very much, and thank you for coming on to discuss it. It was fascinating. And that's it.
Thank you, and thanks for a very enjoyable conversation.
Thank you very much.
Thank you very much.
Until next time.
Time to play the game.
If you enjoyed this episode, don't forget to subscribe and rate this podcast. Also share it with your friends so they can listen and enjoy too and give their feedback. We're open to any comment, any feedback, any suggestions on our email, on our website, comments on YouTube. You can also subscribe to our Telegram channel or follow us on Twitter. Any way you want to contact us is linked in the description. Thank you for listening. Until next time, bye-bye.